Next organelle that I want to mention is mitochondria. Mitochondria is very small, and it's usually seen as the kidney-shaped structure within the cell. Sometimes you can see a microscope through a light microscope, but kind of difficult to say all the time. Mitochondria have two membranes. There's an outer membrane. The outer membrane is conspicuous, and it's more continuous. It's the outer membrane which is all the way out. The inner membrane is the one that it's very long in length because it has a lot of folds and invaginations, and these convoluted shapes they form cristae. Cristae would be the projections inside the matrix, and the purpose of the cristae is to increase the surface area of the inner membrane so that it will help the process of increasing to make energy. For example, liver. The surface area of liver is needed more because there's a lot of metabolism going on in liver cells, which is a lot of things are breaking down, and it needs lots of energy to do that. So cells will actually manufacture mitochondria based on their needs. For example, when you start exercising initially, you may have less mitochondria in your cells, but over a period of time, because you're forcing the cell to metabolize more glucose or to burn off glucose, the cell will actually divide and produce more mitochondria within it. So mitochondria they are the powerhouse of the cell. So as you know that without power nothing can take place, no functions can take place in a cell, in your house even. So the cell needs this mitochondria, and the energy that is produced by the mitochondria is called as ATP. ATP stands for adenosine triphosphate, and we'll do a chapter on the whole process of this powerhouse and how this energy is released. Now, another interesting thing to mention over here is because mitochondria comes as a prokaryotic organelles once upon a time, within the matrix, which is the fluid part of the mitochondria, there are sometimes DNA present in here, and the DNA that is present in the mitochondria is different than the DNA present in the nuclear membrane. So this is one interesting piece. Usually when they're doing fingerprinting and where they're identifying victims, they usually look for the maternal DNA, which is the one that is passed on from the mitochondria. Mitochondrial diseases will slow down metabolism. Other structures are lysosomes, peroxisomes, vesicles, and vacuoles. Got a typo over here. Please fix this. There's no Y in here. Lysosomes, peroxisomes are both your garbage disposal area, if you can think of, or it could be a recycling center of the cell. Peroxisomes are similar to lysosomes, but they are very specialized and they only deal with removal of peroxides. Lysosome, on the other hand, are specialized vesicles that will actually, sometimes it will engulf and break down certain components. Sometimes it will also help in removing toxins out of the cells. Sometimes lysosomes participate in cell death, which is the normal part of development. So lysosomes are your storage area where important things can be recycled, and it's also destroys certain non-functional organelles and proteins that are not needed. The ones that are more specialized are peroxisomes, which are only doing on peroxides. Besides these, there are also present vesicles, and vesicles are membranous sacs that are present, and they also sometimes will be formed as a result of forming proteins or lipids out of the endoplasmic reticulum. And these are sacs that will actually They pretty much will bring substances into the cell that are ingested from the outside. Sometimes vesicles will, there are secretory vesicles which will fuse with the plasma membrane to release a substance outside. So vesicles are regions, or their membrane. They have a membrane in them, but it's not a double membrane. But it does have a selected sac-like appearance, helps things move around, or more like a transport mechanism you can think of. Now, vesicles that are really large are called as vacuoles. Plant cell has a very large vacuole. So if you look at the picture in the book of a plant cell, the big abundant space that you will find are the vacuoles. Sometimes vacuoles are also present with water only, and they will help give tonicity to the cell. Sometimes vacuoles are present with which will store substances, and these are mainly, of course, in plants that are storage. But in animal cells, sometimes usually have smaller vacuoles. We don't really have large vacuoles in animal cells. Now, let's look at the most interesting system of transporting things, and this is the endomembrane system. The organelles in the endomembrane system work together to function many steps that are shown in this particular pictures. So I'm going to begin with showing you how it gets started. The nuclear envelope is considered to be a double membrane, and the outside membrane is sort of continuous, and it keeps on going into the cytoplasm region. And when it keeps on invaginating into the cytoplasm region, it forms many kinds of folds. Now, these folds are called as the endoplasmic reticulum, or ER in short. Some of these endoplasmic reticulums have surfaces that have things that looks like beads studded in them. Let me erase this with a razor, and you can see over here. You can see the yellow regions that are shown that are present on this surface over here. But notice the ones on the bottom over here do not have these studded beads-like structures. The beads are ribosomes. And we will talk later again about the ribosomes. Ribosomes are your protein manufacturing regions. So when ribosomes are present on the endoplasmic reticulum, they will produce the protein, and these proteins are then they're sort of transported through these vesicles into a system which is called as the Golgi apparatus. So one end of the Golgi apparatus will absorb these transport vesicles, and it will pass through these vesicles eventually into, sometimes as secretory vesicles. The Golgi apparatus is like a UPS system, which is it packages and it transports these products that are formed. Proteins are present within these vesicles. Now, the smooth endoplasmic reticulum, on the other hand, helps in manufacturing lipids only. So, if there are more smooth endoplasmic reticulums, then they will synthesize lipid and perform various other functions along with it. And they will, again, the transport vesicles that have lipids also fall into the Golgi apparatus, and they are then transported in a similar way to forming vesicles, which are transport vesicles, to then be either excreted out through the plasma membranes. This is important. Make sure you can narrate this down as an essay type question, like how are these substances formed, which will be in a different chapter. But once they're formed, they are gathered in these vesicles, and they're transported to the Golgi apparatus, where actually the Golgi apparatus also sorts them and packages them into vesicles. Just like if you had to send a package of some glass product to, let's say, New York or some Europe, you would do a better packaging to it versus something that is required locally. The cell does the same thing. If the cell needs a protein locally, it may not synthesize it. It may synthesize it but not package it that well, because it's needed within the cell or to an adjacent cell. So we just talked about ribosomes. What are ribosomes? Ribosomes are little beads-like structures that were shown previously. Ribosomes are actually made up of two subunits. So there's one unit on the top and a unit on the bottom, and it's always the S value is a sediment value of these two subunits. The S value differs for prokaryotes versus eukaryotes. And if you recall, I mentioned about how antibiotics are designed to target the prokaryotic S value particular ribosomes and not the eukaryotic ones. Ribosomes are a very important region. They collect because they're collecting from both the smooth endoplasmic reticulum and from the rough endoplasmic reticulum lipids or proteins. They will package them and they will distribute these molecules to whichever location they are needed to. Okay, now let's look into the cytoskeleton that the cell would have. A cytoskeleton is a component of mainly proteins that are interconnected, and they extend from the nucleus to the plasma membrane in many eukaryotic cell. The cytoskeleton are composed of either actin filaments or intermediate filaments or microtubules. And let's look at all three of them, and you can then deduce as to which are present in which kind of functions. The actin filaments are usually long, and these are present in muscle cells, and these filaments help muscle cells glide. So any kind of gliding movement that you see in the muscle cells are related to actin subunits. On the other hand, microtubules, these microtubules are more like pipes that are like bunch of pipes that are put together into a more tensile or strength, and microtubules are organization that usually comes up when the cell is trying to maintain its shape, or even when the cell has to move substances from one end of the cell to the other, or the organelles need to move. Microtubules are considered to be more like railroad tracks within the cell. These are the railroad tracks, help things to move around. Sometimes microtubules also help during cell division, in which they will form spindles where the chromosomes will align and help it to be separated, and we'll talk about those in the chapter of cell division. And the last ones are intermediate filaments. Intermediate filaments are in the size intermediate between the actin and the microtubule, and they mainly support the nuclear envelope and also the plasma membrane. Intermediate filaments, one of the important function of this is that it helps for the cell anchoring of the cell. For example, skin cell will adhere to each other due to intermediate filaments. So I'm going to write down anchoring and communication within cells. Microtubules are the railroad tracks, helps in movement, and actin filaments also are required for the movement. Well, actin filaments are required for its own movement. Railroad tracks are actually more like transporting material. So that would be this one. Anchoring organelles are this one, and cell movement are the actin filaments, and the rest are still, they all definitely still give shapes to the cell. Almost to the end, the last slide, which is about what are cilia and flagella. Cilia and flagella are both structures that are needed for movement. The difference is the cilia is more hair-like, short structures. For example, cilia can be present all around the mucus lining of the throat, so it will help move particles. It could be present in the nasal tract to sweep debris, and there are many of them. So if you look at a cilia on a cell, the cilia would be short hair-like structures all over. Flagella, on the other hand, in a cell would be just one or two perhaps, and they have a very unique 9 by 2 arrangement, which is a microtubule round a very central spoke like arrangement that is shown over here. And both of them are pretty much used for movement, but it just depends. Flagella will help the whole cell move by itself. Cilia may just help in gliding, and sometimes cilia can also help in adhering to a surface. So this concludes our exploration of the cell and all its structure and its function.